Okay, so for this audio lecture, we're going to be looking at chapter 27, which is controlling microbial growth. So chapter 5, we were looking at microbes and making them grow. And for our last two chapters for this unit, we're trying to kill the microbes. With controlling microbial growth, for this chapter, what we're looking at it is on surfaces. So not treating someone for an infection, but trying to control the microbial growth so we prevent infections potentially. And there's a lot of terminology with like each of these units, but there's going to be some terminology that you're going to want to become comfortable with and know the definitions of. When we look at controlling microbial growth, there's different agents and procedures that can be done, and they fall into the category of physical agents, chemical agents, and then there's these kind of miscellaneous categories, and we'll talk about those in just a moment. When we have a physical, chemical, or other agent that we're using to control microbial growth, we would look at how effective it is. So I always kind of refer to it as this level of clean. So the effectiveness of control can lead to disinfection or sterilization or sanitation. And then within that it could be because the agent is either working cidal in a killing manner or static, and then it just doesn't allow for the colony to grow, increase in number. So with static, it's kind of like you're stuck in this lag phase. So a lot of that's going to make more sense in a couple slides, but just to kind of lay out the topics that we'll be talking about. So this is a very key slide for this chapter because it goes over all the different agents and techniques that can be utilized in order to control microbial growth. So you should know the different categories and then examples of techniques or agents that achieve that can be used as a physical control using heat, that can be used as a physical agent for radiation. Same for chemical, right? So you should have examples, and what's really great is just in our house and also like in the micro lab, you were doing things that are meant to help to control microbial growth. So you have firsthand experience with this. I want to point out in the boxes at the very bottom of each part of the tree, in the black, gray, and white, and then also in the yellow, you'll notice those terms. Those terms are really important, so you should have working definitions of those terms. The next slide goes through and gives you definitions of those, but because we're on this slide, I'm going to go over them. So you'll notice sterilization. So for sterilization, that's where it's devoid of life. So kind of thinking like vampire-like, right? Devoid of life. So really what we're talking about is that viable organisms, metabolically active, but also endospores are destroyed and removed. So the gold standard for sterilization is that even the endospores have been killed so that they're not going to be able to germinate, they're not going to be able to reproduce and then lead to an infection. So it's really devoid of life. We do want to keep in mind with sterilization, again, the gold standard is endospores. There are microbes that are actually more resistant to different methods than endospores. So some of your acellular organisms like Puron diseases, those agents actually are even more difficult to achieve sterilization with if we were using them as the gold standard. And later on in the semester, when we talk about acellular infectious agents, we'll go into that in more detail, but just keep that in the back of your mind. Again, for sterilization, the gold standards, there's no viable cells and there's no endospores that could germinate, could repopulate. You'll notice there's disinfectant and antiseptic. So disinfectants and antiseptics give us the same level of clean. For them, we are making sure that any of the vegetative cells, metabolically active cells, are killed. But we don't it's not that we don't care about endospores, but we're not looking to actually kill endospores with disinfection and antiseptic. And that's because usually in the situation that we're dealing with, we're not dealing with endospore producing bacteria. So there's no point in going to that extra step of sterilization because, again, we don't have endospores present. Again, I'll give you some examples of that in a bit. The difference between disinfectant and antiseptic is that with a disinfectant, it's treating a surface, a non-living surface, right? With antiseptic, it is going to be a living surface, so there's a tissue, there's cells involved that we don't want to kill, like our skin, and so we would use a antiseptic like on our skin because we want to make sure we control microbial growth and don't get an infection because of the cut. So again, we don't want the point where we're sterilizing the skin because we don't want our skin to die because that's alive, right? So again, the criteria, like if we're treating our skin because we got a cut and we don't want an infection, we're looking for an antiseptic. Now, if we have a table surface, maybe where patients are going to sit or, in different situations where you want something clean, but you're not going to do surgical procedures on it, then that's where disinfectant might be good enough because, again, you're not looking to control the endospores, okay? And then in the yellow box, you'll notice chemotherapy. Those are your antimicrobials that we'll be talking about in chapter 28, so just to point that out. There is another level of clean, and that is sanitation. With sanitation, you're just taking it down a notch. You're going down a notch from disinfectant, antiseptic. You're just looking at reducing the microbial population. So you'll notice that none of these actually are showing that. And really, if you were to dilute out concentrations, decrease contact time for a lot of these different techniques, that would actually get you to sanitation. And I'll point out an example of sanitation during this lecture too. So, all right, so we're going to kind of walk through this chart, and I'm going to point out some key things. So you'll notice under physical agents, we have heat and radiation. So with radiation, there's different types of radiation: ionizing radiation and non-ionizing radiation. Non-ionizing radiation would be UV light. So UV light actually doesn't penetrate deep into tissues. That's one of the reasons why when you're exposed to UV light from the sun, you end up getting like skin cancer, but it doesn't cause internal cancers, right? So that's a way to kind of keep that in mind. So we use UV lights to disinfect because, again, they can't penetrate, they can't sterilize. It's only just like this kind of surface treatment. We use UV lights in hospital situations. There's UV robots that will go into the patient's room and expose the room to UV light. Also in the research setting, your hoods that you would work in for tissue culture, they have a UV light. So when you're not working in the hood, you would turn the UV light on. Ionizing radiation has a deeper penetration, so it can go into tissues, and it can damage the DNA in a way that would cause the cell not to be able to replicate. Remember that microbes usually are single-celled organisms, like especially when we're talking about bacteria, single-celled organisms. So if that one cell gets mutated, there's not the other cells to come in and repopulate. So X-rays and gamma rays, those are examples. And using those, you could actually achieve sterilization. So if you've gone through customs, your suitcase goes through an X-ray machine, and part of that is to determine if you're smuggling anything in, but also that can help to sterilize any kind of potential microbes and spores and such that you might be bringing in unknowingly, or because you decide to smuggle some fruit in. So that's always an area of concern where we might introduce pathogenic organisms into another area that could decimate like the agricultural industry. So that's why when customs, they'll ask you, did you bring any fruit in, or because there's potential that they could bring in some kind of contamination. So important that is sterilized. Then looking at heat, you'll notice that it splits because there's dry heat and moist heat. So I always like to give the example of where do people retire? They retire to dry climates, right? So like Arizona, Florida, where it's not so humid. And they'll tell you that the dry heat just doesn't feel the same as like when it's all humid. And you can even think about, here on those humid days, you don't want to go out, you're a lot slower moving around. But when it's nice and cool and low humidity, you're like, this is great, right? There's a difference between when there's water in the air and those water molecules are bombarding you with that humidity. It just wears on you. Well, if you're a single-celled organism, that water can damage you, and then damage you in a way that you can't reproduce, or that you are actually killed. So with dry heat, because there's not water molecules present, the temperatures that are needed are going to be higher in order to achieve sterilization. So you'll notice incineration is listed in dry oven. So dry oven, you're going to have to expose to relatively higher temperatures for long periods of time. So think about like if you were to leave something in your oven at like 400 degrees, your oven's going to be relatively dry heat. You can achieve sterilization, right? With incineration, instead of being 400 degrees Fahrenheit, we are then turning this up to like 600 degrees Celsius or higher. So in the lab, you're going to be using Bunsen burners, ethanol flames. That flame, that blue part, the very top of that blue flame is about 600 degrees Celsius. That's wicked hot. So that's why you put your loop in there, because your loop will heat up and any kind of organisms on it are going to be incinerated and burned, because 600 degrees Celsius is too hot for them and it will sterilize them. When we're looking at incineration of like biohazard material, those incinerators are going to be a lot hotter. So they're anywhere from like 1,000 to 2,000 and above, depending upon the type of incinerator it is. So it's possible for medical clinics to have on-site incinerators that might be the size of like a microwave. Those are not going to be as hot as commercial incinerators, like for companies like Stericycle. They will haul away biohazard material, and then they have incinerators that are the size of buildings. So those are going to be a lot hotter. Incinerators are in hospital settings. So many hospitals have their own incinerators on site, usually on the top floor, because heat rises. Let it start at the top, right? Also for safety reasons, it's also better to have it on the top versus on the basement floor. So with those incinerators, they're not going to be as hot as Stericycles because it's within that building, okay? But again, it's going to be a lot hotter than your Bunsen burner or even that smaller, like microwave-sized incinerator. All right, so again, with incineration, you can achieve sterilization. So that is why that technique is used in hospital settings, because the surgical that biohazardous waste has a high probability of having infectious material on it. All right, we're going to look over at moist heat. So with moist heat, again, we have these water molecules that can help damage the membranes, denature proteins, so that these enzyme systems of the microbes can't work, so they can't reproduce, and then they die. So with moist heat, what you might be familiar with is if there's been a lot of rain and there's a lot of runoff, and then that potentially is going into like drinking water, they might have a boil order where you have to boil your water to make it safe for you to drink. Or if there's been a pipe that's broken, they might ask you to flush your pipes, run the water, but then you might have to boil it for 2-3 days. They're going to tell you to boil the water. Water boils at 100 degrees Celsius, and they would have you boil it for 30 minutes. So that 100 degrees Celsius for 30 minutes is only going to achieve disinfection, but that's okay, because usually what they're looking at is that there's coliforms, such as like E. coli. So these are fecal bacteria. And so the concern is of these fecal bacteria that do not produce endospores. So sterilizing the water is really not necessary because, again, there's not going to be those endospores present. Now, in a hospital setting or in a research setting, it's possible, or in our educational labs, we might be working with endospores and we want that material to be sterilized afterwards. And so we're going to want to achieve sterilization. Well, it's going to be pretty hard to do that with just boiling material. And so what you do is you actually put it under pressure. And when you put it under pressure and you have that moist heat, water, and you heat it, you're going to have steam. But that steam and that temperature that you achieve is actually 121 degrees Celsius. The 121 degrees Celsius, that little difference of 21 degrees Celsius, makes it so that you can achieve sterilization. So 30 minutes at 121 degrees Celsius can achieve sterilization. The way to do that is by putting it into under pressure. So you might recognize like a pressure cooker at home. So if you think about preparing a meal, if you use a pressure cooker versus your oven, those temperatures you use are different and the amount of time are going to be different, right? To cook, to denature the food so that it's edible. In the research or hospital setting, steam under pressure would be autoclaving. So autoclaves are used to sterilize surgical equipment. It is important that the material that you are using can be autoclaved, that it can survive that higher temperature. So there's certain like suture material is not going to hold up well. Certain plastics don't hold up well. So if we're going to use those things, we need to have other ways in order to control the microbial growth on them. Okay, but autoclave helps with that. There's special plastics called Nalgene. So before Nalgene bottles were all popular in the mainstream, Nalgene was the hit thing for scientists. So you'd have to have Nalgene trays and then Nalgene bottles where your solutions were put into it. So just like incinerators come in different sizes, autoclaves come in different sizes. So there's tabletop autoclaves, and then there's larger wall to ceiling type autoclaves, and then there's roomful autoclaves. So depending upon who is using it, what environment, whether it's a research environment versus hospital environment, changes kind of like what size they would purchase, and depend upon how much material has to be autoclaved. Towards the middle of the sphere, you can see chemical agents. If you follow that down, chemical agents fall into two categories: gases and liquids. So just to give you examples of those, with gases, one of the common one is ozone. So ozone is a gas that's based on oxygen. So that is used in the egg industry. So they actually have these chambers that they'll put the eggs in, and then they fill that with ozone, and that actually kills any kind of surface bacteria that's on that egg. And so it can be used depending upon contact time, concentration, for disinfectant or sterilization. You can also take hydrogen peroxide and put it into like a gas form. And I'm sure like someone would argue like, well, it's a little bit more like liquids, but we've just made the particles really small. Another example of a chemical agent would be hydrogen peroxide, and that's also utilized both in home setting, but also in like hospital setting. So at home setting, you would use like a 3% hydrogen peroxide, either as a disinfectant, antiseptic. In the hospital setting, they would use a higher concentration, like 30% to 60%, and that can actually reach sterilization. And again, there are apparatuses and robots that use hydrogen peroxide, and they actually put it into like the air, And then that goes over any of the contact spaces and can reach sterilization. The reason the robots are used is because there's a potential toxicity because it's a very high concentration of hydrogen peroxide. And so by using a robot, you don't have to have anyone being exposed to that in the room when that's being utilized. With our cleaning product activity, you guys saw lots of different liquids. So you guys went into your cleaning cabinet and looked to see what you had, and you guys had lots of different examples of active ingredients. A lot of them were ethanol and chloride, halogen based products. And so again, you can see how there are different chemical agents that are used in a liquid form, and also within that, some of those were disinfectants, some of those were antiseptics. In the home setting, most of the times they're antiseptic or disinfectant. Not usually going to find something that's to the level of sterilization in your home setting. Now, when we look at hospital grade liquids, a lot of times what they've done is increase the concentration. There are certain chemicals that they're more likely to get access to, and so that can really reach sterilization. So with that, just in that distinction of your home setting versus the hospital setting, we want to keep in mind in your home setting, when you're buying these like at your local store, right, you could be using that every single day. So part of it is that we want to make sure that the concentration of that is something that if you're exposed to it every day, it is not going to be toxic to the point that it actually kills you. In the hospital setting, the patient's coming in like once, twice. Like for most of us, we might have 2 or 3 hospitalizations in our lifetime, but we're not spending every single day in the hospital. So the toxicity issue is not really with the patient, it's with an employee. So an employee is going to do day in, day out. They get some vacation time, PTO, and all that. But and if they're going to have a career there, I mean, they might be spending 30 years. Maybe they're really smart and they retire younger. But we're potentially talking about someone who could be exposed to this chemical for like 30 years, right? And even if they're not staying at that institution, they might be going to another institution. And again, so we want to make sure that we're protecting the employee. So a lot of times when hospitals are selecting the chemicals they are using, again, it's not the patient they're thinking about, it's actually their employees and what is the possible toxicity with that. And again, you see a lot of uses of robots being used to eliminate the potential exposures to employees when the toxicity risk is the highest. So I wanted to point that out. Okay. On this figure, you'll notice in the green, mechanical removal methods. So there's different filtration apparatuses, devices that can help to remove microbes. So both from the air and also from the liquids. So I will point out that filtration doesn't always lead to sterilization. So depending upon what is the filtering membrane, the level of clean, the effectiveness might be different. You might only get a disinfection or a sanitization, okay? So looking at examples of filtration with air, that would be like your HV system. So your heating, cooling system, there's gonna be a filter on that. So it's gonna filter out large particles. If you have a nicer filter, microbes, some microbes like bacteria, and then if you have a really fancy filter, it has a charge to it, and so it can actually filter out viruses from the air. But at no point would I consider that sterilization, just because in a host setting, it's just the dynamics in airflow doesn't allow for sterilization to occur. If we move into a lab setting or hospital setting, their buildings are set up so that there's like a negative flow, so everything flows into the HV system. They also have so many rates of exchange in a given time frame, and so in those cases you can reach a sterilization. Their filters are also different, so they have HEPA filters, HEPA, and so those HEPA filters actually are able to filter out all material, hence why they're used in BL2 and above labs, because potentially you're working with infectious agents that are more infectious and more prone to causing infections, and also obviously in a hospital setting. Now, when we're looking at filtering liquids, this might be where we are in the lab and we have a solution and we want to make sure we're not contaminating our cells with bacteria. So when we make our media, we would pass it using a vacuum through a membrane. That membrane can be different, the holes can be different sizes, the pores can be different sizes, so depending upon what we want to filter out, we could use different ones. Sometimes those filters have a charge to it, so because viruses and other particles have charges, it would capture those. Okay, if we were trying to isolate viruses, we can filter them and trap them on membranes, and then they're on one side and our other stuff is on the other side, sterilized. Sterilization of liquids in the medical fields are important because there are certain drugs that are in a liquid form that then get injected into patients and can be injected into their nervous system, their central nervous system. And so if those aren't sterilized, that can obviously lead to complications. And so it is really important, like in a compounding pharmacy or a general pharmacy, if they're making up these solutions, that they are using sterile protocols. So they're doing this in a sterile room, taking precautions to make sure that they're not introducing their own microbes, making sure that room is truly a sterile room, so there's no like dripping pipes and everything and mold growing. And there's been cases where there's been pharmacies that haven't had this, and it's led to really negative outcomes. And I talked about that a little bit in class. Okay. All right, so we're not going to actually talk about the biological agents for microbial control. There's enough stuff to talk about. All right, so that really talks through the whole entire chapter, so that one figure. So I'm going to go through these slides really quickly. So when we're looking at the level of clean or the effectiveness, you're going to hear terms like sterilization, disinfectant, antiseptic, sterilization or sanitation. So again, we have different examples of sterilization where the material, the surfaces become devoid of life. So that's where we're removing the vegetative cells and also endospores. Disinfectant, antiseptic, we are removing the vegetative cells, so we're ignoring the endospores, or there's no endospore producers present, so that level of clean is sufficient. Okay, disinfectant is going to be on a non-living surface, where antiseptic is going to be on a living surface. So we want to make sure that we have to keep in mind that we don't want to damage that living tissue. We just want to damage the microbes. And then sterilization is a reduction of the microbial population on a surface so that it's then safe. So my example of this is if we think about cleaning the toilet bowl. Most of us actually just sanitize our toilet. So we might use a toilet brush and scrub it. And that scrubbing actually is a mechanical removal of the microbe, because the movement of scrubbing the toilet reduces the number of bacteria that are present because you dislodge and disrupt those biofilms. And so then in disrupting those biofilms, you end up removing those microbes from being present. Okay, that's sterilization. Now, you might throw in some Mr. Clean or scrubbing bubbles or something. So you might get to the point where it's disinfected, where you've killed all those vegetative cells. Probably not, because you probably didn't get every little nook and cranny. And that's okay. So sanitation for your toilet is fine, right? That's going to remove the microbes. It's going to make it safe for you to engage in normal activity with that toilet, which is sitting on it and doing your thing. The reality is we're not looking to eat from our toilet, right? So maybe your dog does, but you're not. And so that level of disinfection really is not necessary, and definitely not sterilization. So we always want to look at what are we using the surface for? What is the level of clean we actually need to engage in normal activities, okay? So again, sanitation is actually fine for your toilet. When we're looking at something like your kitchen, this is where disinfection is usually the level of clean we need. So most of, when we're looking at foodborne illnesses, not all of them, but most of them are due to non-endospore formers. And so if we think about Salmonella, E. coli, Listeria, disinfection is that level of clean that makes it safe for us to be working in that surface and eating food prepared in the kitchen and such. Now, if you were performing surgery in your kitchen, which I do not recommend, I'm pretty sure it's illegal, then sterilization is what you're looking for. But again, if we're not cutting into someone, we really truly don't need sterilization for most cases. Okay, so we always talk about like sterilizing our cutting boards and everything. Like, one, we're not technically probably actually reaching the level of sterilization, and two, again, most of the organisms we're looking at causing illness that are from our kitchen do not require sterilization because they do not produce endospores. So again, you always want to kind of think about like, am I going too much? Because remember, to reach sterilization costs time and money. So disinfection and antiseptic are quite more affordable from a resource standpoint of money and time. All right, and then keep in mind that there's also the chemotherapies, antimicrobial agents. When we look at the mechanism of how some of these controlling of microbes, but also with antimicrobial agents for our next chapter, there's the terms cidal and static. So cidal refers to killing. So you might see something, if you look at your cleaning product, you might notice that it's bactericidal or microcidal or fungicidal or virucidal. That indicates that the mechanism actually kills the bacteria. If it says static, like bacteria static, fungal static, it just inhibits the growth. So it doesn't allow for the population to grow. It just keeps it the same level. Static agents have the possibility of killing, but it's killing because it's depriving them of the ability to reproduce, and eventually cells just die, okay? So when you look at your cleaning product, you might notice it says it's microcidal. Again, that would indicate it actually kills. Most companies don't advertise like, hey, we're bacterial static. So that's something you see more with mechanism of action for antimicrobial agents. So we want to keep in mind with microbial death, killing of those microbes, they're not always killed instantly. Sometimes it's over time. Okay, that's kind of the gist of this slide. And keep in mind that we're looking at viability. So we want to achieve microbial death. We want them to, it's not that, it's possible that they're still viable, but they're just not reproducing, right? So that's where we get the static. All right, so I'm going to go through these slides pretty quickly because we've already talked about these, but this gives you an opportunity to see the figures that actually show you the different techniques. So when filtration, you can see here with this membrane that's shown kind of in the pink, there's holes in it, and so the bacteria can't get through the holes. So that means when we have our apparatus set up, which you see in panel A, the fluid is going to go through, the liquid's going to go through, but the bacteria get trapped on that filter, that membrane, and so what we end up collecting is sterilized fluid. It's not, I always struggle with this one. So it's not usually technically sterilized because sometimes, again, you want to keep in mind some of these membranes do allow viruses to go through. So viruses technically aren't living. So it's not really that it is sterilized because it's devoid of life because, again, viruses aren't living. So that's a whole debate. We can also filter air. So here you see this. This is a fume hood that this individual is working under, and so that you can see the HEPA filter there that's going to filter out the air. With moist heat, again, we have water molecules that can do work, and so temperature does matter. So you'll notice here that if we have vegetative cells, you'll notice those temperatures are like 60, 70 degrees. If we have endospores, if we have spores, we need to be at a hotter temperature. So boiling tends to kill the vegetative cells so we can get disinfection, but you need a higher heat, a higher temperature in order to get sterilization because of those spores. So again, that's where it's really important. You'll notice with bacteria that if we can get it to 121 degrees Celsius, we only need 12 minutes. Now, standard protocol is to do 30 minutes to really make sure you kill those endospores. But that's why we want a closed system, like what you see here, where it's under pressure, and that being under pressure allows the autoclave to reach 121 degrees opposed to being 100 degrees. The pot you're boiling on the stove, it's not a closed system, so that water can only achieve a temperature of 100 degrees before it becomes steam. But if you close the system, you can get it to a hotter temperature. We can also use dry heat, as I mentioned. So you can see here, if we do a temperature between 160 and 170 degrees Celsius for 2 to 3 hours, it is possible to get sterilization. This is really great for things that can't survive really high temperatures, or like chemical exposures might corrode them. And then with radiation, we had mentioned UV radiation. So again, keep in mind that this is more a surface treatment, so it doesn't actually penetrate through things. And so it actually causes these thymidine dimers. So thymidine being the T nucleotide. So that's going to actually prevent replication and transcription. So the cell won't be able to grow and divide. Where ionizing radiation is going to actually penetrate deep into the objects, and so it actually causes breaks in the DNA, and so we could reach sterilization with that. Just to point out, with the non-ionizing radiation, with those thymidine, you're not killing the cell. It's just when the cell goes to reproduce, it can't reproduce. So non-ionizing radiation would be an example of static. Ionizing radiation would be an example of cyto. All right, and then the next series of slides look at chemical controls. So again, remember, for most of these, they fall into the disinfectant or antiseptic. What makes them then sterilization is a higher concentration or increased contact time. So there are phenol-based products. So a lot of times laboratories and laboratory and hospitals will use these for disinfectants. These don't tend to be the ones we use in household settings, but they actually will denature proteins and disrupt cell membranes. For alcohols, these are very common in household products. So many of you guys saw these when you opened up your cleaning cabinet. Most of them are ethanol-based, and these can be cyto, but they are a disinfectant, antiseptic. They don't kill endospores or spores, so they're not sporicidal, so they wouldn't give you that level of sterilization in most cases. And then we have the halogens. So the halogens, if you're looking on the periodic table, there's the column that include iodine, chlorine. So anything on that can act as a halogen in an antimicrobial way. So iodine is utilized. So this at higher concentration can actually kill endospores. So it ends up disrupting proteins and oxidates different cellular components. It can damage your skin and stain, and some people have allergies to it. But when surgeons scrub in, they use an iodine wash in that scrubbing process, and that's meant to help to sterilize. Doesn't truly sterilize their skin because it doesn't kill their own cells. So it is actually more of just a high-level disinfectant in that scenario. With chlorine, it's going to work in the same kind of mechanism as iodine. So it disrupts the cell and its components. Chlorine is able to disinfect, is utilized in like bleach, and so water supplies, swimming pools, you see applications of that. And again, when a lot of you looked at your products, they were chlorine based. So it tends, at the lower concentrations, it is going to achieve disinfectant, antiseptic. High concentrations, like again used in like a hospital setting, can actually achieve sterilization. There are also metals that are looked at for their use as an antimicrobial. So because these are ions, they will actually, if they're taken up by the bacteria, it causes them to unable to chelate and affects their enzyme systems being able to function. So this leads to those cells actually being killed, and they can't survive on that surface. So these can be toxic to us too, if they're not actually like plated. So some of the different metals that are used are mercury, silver, arsenic, zinc, and copper. So if you know someone who's much older, they might have had like a silver filling put in, because that silver, like in their tooth, so that silver inhibits the growth of the microbes because it's an ion, and then it gets chelated and the bacteria can't persist. Cavities are due to bacterial growth, so again, it makes sense to do that. Now, that becomes very expensive, and there's potential toxicity of having that. So again, you see there's been a shift of using different material in order to do cavities, and just making sure that the area of the cavity is cleaned out and treated other ways to minimize growth. Okay. But you can see silver being used in other applications. You can actually get bandages that have silver embedded into them. There are creams with silver, so burn patients are susceptible to infection because they have a break in the first line of defense. And so that cream has silver in it, so the cream kind of soothes the pain related to the burn, but then the silver helps to decrease the microbial growth or chance of infection also. Copper, gold, silver is also looked at for surgical settings, especially in MASH units where you have these mobile units that are being set up in places of war. So soldiers get injured and they have to have surgeries done like right near the battlefield, and so there's a high probability of infection, and there's not always the ability to get material in or time in order to treat surfaces to decrease the chance of infection. So their Department of Defense was looking at using metals to actually line some of these surfaces of these mobile units, like the tables and such, to help decrease the chance of infection in these individuals. So metals are actually a very interesting area that people are looking at. We also have the aldehydes, so formaldehyde and glutaraldehyde. So these will actually cross-link proteins, and we'll see this kind of in the next slide. Because they cross-link the proteins, it means that the cell cannot move said proteins, because cells are made up of proteins. It kind of freezes it. It can be sporicidal, but in some ways it's kind of static, so it doesn't directly kill it, but it's this inability to move the proteins that causes the cell not to be able to reproduce. Aldehydes tend to have high toxicity, so they're only used really in rare cases. These are the same things that are used to fix tissues. So if you have like a biopsy done, it would be put in formaldehyde to fix it so that that time that elapses, those cells can't change and alter. So again, there's toxicity to it. So again, it's only used in really rare cases. So there are certain plastics that don't hold up well to like autoclaving and also other chemicals. So they can be treated with formaldehyde. Those plastics sometimes are used for breathing tubes. Most people don't have a breathing tube in long term. Like the breathing tube might go in, they're going to have it in for a given amount of time, and then it gets taken out and they recover or they don't. So the exposure to formaldehyde is going to be more short term, and the concentration is going to be low. So that would be an acceptable use. Here just shows you that cross-linking between the proteins. We also have gases that can be used. So I'd mentioned ozo, hydrogen peroxide also. They can be utilized to sterilize material. So you might have a chamber. So you see this nice little chamber here that they put stuff into, and then the chamber fills with the gas, and that kills any of the bacteria that's on there. So products that are used for antimicrobial in different applications, home and also in hospital, medical settings, etc. There's 2 agencies that regulate them. So there's the Environmental Protection Agency, the EPA, and there's the Food and Drug Administration, the FDA. The FDA specifically regulates agents that are going to be consumed, used on humans and animals. Okay. And as I've kind of mentioned throughout this audio lecture, there's a number of different things that influence antimicrobial agents' activity. So population size matters. So obviously, if we have a larger population size, it's going to take longer to actually kill than small populations. So if you clean that toilet every single, day, week, whatever, it's going to take a lot less energy than if you only do it like once a month, right? So same thing like on these surfaces that we can't see that build up. If those are cleaned regularly and there's a smaller population, it's going to take a lower concentration, less time exposure for you to achieve that level of clean. The population composition matters. So as I mentioned, sometimes we don't have endospores producing bacteria present. So again, that level of clean that we need, the effectiveness is changed. The concentration of our agent matters. So as I mentioned, in a hospital setting, because maybe we're looking to achieve sterilization, we need to use a higher concentration. That higher concentration is going to kill more quickly. So time is money in a hospital setting, where a lower concentration may not give us that sterilization or require a longer exposure time. Contact time matters, so how long that agent is in contact with those organisms will affect efficiency. So in class, we looked at hand sanitizer and contact time, and I had mentioned how, to kill E. coli with hand sanitizer, you need 3 minutes of contact time, and most people found that that hand sanitizer had evaporated in 15 to 30 seconds. So you're probably not getting 3 minutes of contact time to kill E. coli. And E. coli is a coliform. It would be something you might pick up in the porta-john or in a bathroom. And so, again, we want to keep in mind if we don't get sufficient contact time, we're not getting the level of killing we probably are thinking we are, or that is advertised on the product, okay? And I forgot to mention this in class, but keep in mind that it's the active ingredient needs to be tested. The product is not required to be testing. There were guidelines from the FDA suggesting that the actual product should be killed to make claims, but again, the current law is that the active ingredient has to have that level of killing under some kind of testing situation, which obviously between the active ingredient and the actual product, there can be lots of variations. As we mentioned, temperature, so looking at heat, the higher temperature is going to achieve a higher level of microbial control. And then there's also the local environment, so what's the pH, viscosity, are there biofilms present? So all those can play a role. So if you do have any questions, please let me know. I'm more than happy to answer your questions. But hopefully that provided you with the background information on controlling microbes. So thank you.